Hello everyone. This is our session 4 of biological classification. In the earlier sessions, we studied about biological classification. Further on, we proceeded about kingdom Monera, and then we studied about kingdom Protista. And our today's agenda of study at first will be recap, then we'll study kingdom Fungi. So let's begin. So, if I talk about earlier sessions, we studied about biological classification, which was given by R.H. Whitaker. R.H. Whitaker in 1969. R.H. Whitaker in 1969. There were 5 different parameters which he used for classification, which included cell type, cell wall, nuclear, membrane, body organization, and at last we studied about the mode of nutrition. So these were the 5 parameters on the basis of which he classified different organisms. Further on, when we proceeded, we came to know the 6 kingdom classification, which have had 3 domains, and the 5 kingdom classification given by R.H. Whitaker have 2 domains. Further on, we have our kingdom Monera, and in kingdom Monera we studied about different bacteria. We studied about archaebacteria, we studied about eubacteria. Kingdom Monera. We talked about archaebacteria and eubacteria. If I talk about archaebacteria, they have an extremely resistant cell wall towards extreme conditions, which is made up of glycoproteins. And further we studied that eubacteria have a rigid cell wall. Archaebacteria examples are methanogens, found in marshy lands and gut of ruminant, can be used for biogas production. Further up we have had our halophiles, at number 3 we have had thermoacidophiles. Phile means loving, so halophile, salt loving, thermoacidophile, temperature, so hot sulfur springs. Further we studied about the eubacteria, and in eubacteria we studied about cyanobacteria, cyanobac, and gram-positive, gram-negative bacteria. If I talk about cyanobacteria, they have similar chlorophyll A as that of higher plants. Chlorophyll A similar to that of plants. Therefore, they are photosynthetic. They are photosynthetic autotrophs. Autotrophs is that they cook their food by on, and photosynthetic with the help of light. And further on, we study about the cyanobacteria. We have nostoc, Annabella, and oscillatoria. So these are the examples of bacteria responsible for nitrogen fixation, and they have specialized cells called nostoc, called heterocyst, into fixing cells. These bacteria are generally covered by a covering. This covering is mucilaginous, and this is heterocyst. Further on, we studied about heterotrophic bacteria, where heterotrophic bacteria, where we studied about the curd-setting bacteria, that is lactobacillus. We studied about nitrogen fixing bacteria, free living nitrogen fixing bacteria, that was rhizobium. We also studied about medicine forming, that was streptococcus, used for making streptokinase. And further on, we studied about our chemosynthetic, which use chemicals to derive energy for ATP formation. Chemosynthetic bacteria. Chemosynthetic bacteria utilize energy from nitrites, nitrates, and and ammonia for ATP production. For ATP production. Moreover, they are also involved in nutrient recycling, and in nutrient recycling we have nitrogen, phosphorus, and sulfur and iron. So these are the things that it usually nutrients that are recycled, and we have some other bacteria as well known for causing disease to both crops and humans, which includes citrus canker, cholera, and we have tuberculosis, we have typhoid, and there are others as well like crop citrus canker. So that's what we all studied about our kingdom Monera. We also studied about mycoplasma, which can even survive without oxygen, one most important point, and it lacks cell wall. Further on, we studied about kingdom Protista. Kingdom Protista. There we studied about five different phylums, which included number one, chrysophytes. There we have had desmides, pollution indicators as they grow in clean water only. And they are golden algae. They are regarded as golden algae. Then we have diatoms. Diatoms have two thin overlappings, one over other. At first they are chief producers in ocean. Number two, they have two thin overlappings fit together as in a soap box, and which is embedded with silica. So you can say that the soap in the soap box is silica, so which makes it indestructible. Presence of silica makes diatoms indestructible, thereby forming diatomaceous earth. Diatomaceous earth used for polishing, filtration of oils and syrups, and at last it is also used for shining. Yeah, it is used for shining things. That's it. We have studied about chrysophytes, then we studied about dinoflagellates having different colors based on the pigment present: blue, brown, yellow, green. So these are the different types of dinoflagellates that we have. Then further on we studied about they are known for causing red tides. Gonulax is the example. They have an outer surface of cellulosic cell wall. They possess two flagella, one longitudinal and one transverse. Further on, we studied about they are found in majorly marine habitat. However, chrysophytes are found in both marine and freshwater. And as they cause red tides, they are also responsible for causing death of various marine organisms, as they release certain toxins harmful for marine animals as well as fishes. Further on, the third was euglenoids and then slime molds. Euglenoids are found in fresh, stagnant water. They are on dead twigs and leaves. They are photosynthetic majorly photosynthetic in presence of they're majorly photosynthetic in presence of sunlight, and heterotrophic in absence of sunlight. Sorry, if I talk about slime molds, they are saprophytic in nature, as they feed on dead and decaying matter. They feed on dead and decaying matter. They are saprophytic. Number 3, if I talk about, you know that euglenoids are photosynthetic, therefore they possess chlorophyll A. Chlorophyll A, same as that's present in higher plants. Their cell wall absent, present. In place of cell wall, they have proteinaceous, flexible layer called pellicle. They don't have anything like pellicle. If I talk about examples of euglena, we have further on. If I talk about slime molds, there are two conditions under favorable conditions. Under favorable conditions, they form an aggregate known as plasmodium. During unfavorable conditions, they form spores having true walls, therefore extremely resistant. They are extremely resistant and are dispersed in air currents. Okay, so that's it about slime molds. Now we are going to study about the last thing that is protozoan. If I talk about protozoans, we have flagellated, like trypanosoma, known for causing sleeping sickness. If I talk about amoeboid, which have silica shells, spread their false feet, or pseudopodia, for engulfing materials, and examples are amoeba and entamoeba histolytica. If I talk about ciliated, they have a gullet or body cavity where water laden along with food is stirred. So whenever any cilia organism like Paramoecium intakes food, the water that is also enters the body is stirred into the gullet. And at last we are going to study what sporozoans. They have infective stage in their life cycle. स्पोर्स मेजरली पैरासाइट्स होते हैं. एग्जांपल है इसका प्लाज्मोडियम. That's it. Now let's move on to our today's session. That is, we are going to study about fungi. यहाँ पे इंट्रोडक्शन के लिए कोई चीज नहीं है क्या? Now we are going to study about kingdom fungi. Kingdom fungi. So earlier we studied about single-celled organisms. In the first kingdom, Monera, we studied about unicellular prokaryotic organisms, single-celled organisms with primitive nucleus. Further on, we moved ahead with single-celled organisms with well-defined nucleus, and now we are going to study multicellular organisms. Cellular organisms with well-defined nucleus. This is kingdom fungi. They have cell wall made up of chitin. And chitin is a polymer of N-acetyl glucosamine. N-acetyl glucosamine. Further on, we can study about their mode of nutrition. That is either saprophytic or parasitic, majorly saprophytic. Saprophytic and parasitic. Then we can study about nuclear membrane. Yes, it is present. Nuclear membrane. It is present. So if I talk about the body, if I talk about the body, they have long thread-like structures. They have long thread-like structures which are known as hyphae. Long thread-like structure called hyphae. And the network of these long thread structures, the network of these hyphae or these long thread-like structure network, when these hyphae or long thread-like structure form a network, that is known as mycelium. That is known as mycelium. And mycelium can be of two types. Mycelium can be of two types. Okay. We have one with coinocytic hyphae. So coinocytic hyphae are continuous tube filled with multinucleated cytoplasm. That is coinocytic hyphae. If I talk about other sort of hyphae, they are branched. and septate. These are septate and branched. So these are the two types of hyphae which are present. At first we studied about long thread-like structure, which is known as hyphae. Then we studied about network of those hyphae, which is known as mycelium. Then we studied about conoceric hyphae and the branched and septate hyphae. And now we are going to study about next thing, that is the association. So algae and fungi together form lichen as association. Algae is phycobiont as it provides food. Algae is phycobiont. Fungi is myobiont as it provides shelter. Lichen, pioneer species. SO2 pollution indicator. As it doesn't grow in SO2 rich regions, and it is the pioneer species. Pioneer species means the first to come into a land which is where there is no civilization, or there were a civilization earlier there. So that is pioneer species, which grows fast. And then we can study about mycorrhiza. Higher roots of plant and fungi's association. So that is known as mycorrhiza. Next thing we talk about is the reproduction. So as I have talked earlier as well with you about fungi, algae and protonema of mosses, fragmentation. So fragmentation is found in fungi, algae and protonema of mosses. So the first mode of reproduction we can write is fragmentation. The first mode is fragmentation. Number two is fission. Number three is budding. These are the vegetative modes. These are the vegetative modes of reproduction in fungi. Then we talk about asexual reproduction. Asexual reproduction, which is carried out by angiosperms, conidia and zoospores. Next we talk about sexual reproduction. Is carried out by oospores or ascospores or basidiospores. And fusion of two motile or non-motile gametes. Fusion of two motile or non-motile gametes is called plasmogamy. Number two, we have karyokinesis. Fusion of two nuclei is called karyogamy. Fusion of two nuclei is called karyogamy. karyogamy. There is an intervening stage in basidio mycetes and ascomycetes, and that phase is known as dikaryophase, and the condition is dikaryon, where two nuclei are present per cell. So now we are going to study about that, and meiosis is what responsible for the formation of zygote of haploid spores. Results in haploid spores. So meiosis, as when fungus reproduce, at first there will be fusion of two haploid cells, fusion of two haploid cells, which results in the formation of a twin cell. However, in some cases, there is an intervening stage. Like there is N, so the new formed will be N plus N for some times, like N. A single cell with two nuclei. This is an dikaryon. So dikaryon is nothing but is an intervening dikaryotic stage where a cell have two nuclei. Here the nuclei is diploid and one, and here there are two nuclei which are haploid. Okay, so this is the general case in phycomycetes. And if I talk about dikaryon, it is found in Basidio and Ascomycetes. We are going to study these terms further, so don't worry about this. And further on, we study that is dikaryon. There is an intervening stage where two nuclei are present in a cell, and the stage is known as dikaryon, and the phase is dikaryophase. Further on, the parental nuclei fuse and the cell becomes diploid. When those two nuclei fuse, then the diploid stage comes, which is the similar as the earlier found in ascomycetes. Now the fungi form fruiting bodies by meiosis. As a zygote is formed, now the fungi starts forming fruiting bodies, leading to the formation of haploid spores. Now we have zygote, like after dikaryon phase. Okay, let me tell you. First it's plasmogamy and karyogamy. So after karyogamy, there are two things that are possible. N plus N and directly to N. This is the condition in phycomycetes. This is the condition in phycomycetes. And if I talk about N plus N, this is in ascomycetes, and basidiomycetes. So this N plus N intervening stage, when the parental nuclei fuse, it forms 2N similar to this, and this stage is called dikaryon and phase is called dikaryophase. And after the formation of this zygote, what happens is, now it forms fruiting bodies, meiosis occurs, zygote form fruiting bodies, which undergo division to form spores which are haploid, to form haploid spores. And fungi are classified on the basis of morphology of their mycelium, mode of spore formation, and fruiting bodies. This was a question asked this year. Morphology of mycelium, fruiting bodies, and mode of spore formation. This is the basis on which fungi are classified. Fungi are classified on the basis of morphology of their mycelium, mode of spore formation, and fruiting bodies. Let's begin with the phycomycetes. Phycomycetes. So, the most basic classification of kingdom fungi, I'll say. Kingdom fungi. Now we are going to study the phylums of kingdom fungi, which includes phycomycetes. Then we have ascomycetes, the sac fungi. We have basidiomycetes, club fungi. At last we have deuteromycetes, fungi imperfecti. That's what we have in kingdom fungi, and now we are going to study about phycomycetes. We have studied about the phylums of kingdom fungi, which included phycomycete, ascomycete, basidiomycete, deuteromycete. Okay, so now we are going to study about phycomycetes. Phycomycetes are majorly in aquatic habitat on decaying wood in most moist and damp places. Okay, so phycomycetes habitat, they are mostly aquatic and present on damp places. Decaying wood, and P for parasites. They are obligate parasites on plants. Further on, we study about the spore formation. Spores can be of two types: asexual spores and sexual spores. Asexual spores are of two types: motile and non-motile. Motile spores include zoospores, and non-motile are aplanospores. And if I talk about the sexual reproduction, zygospores. Further on, we talk about mycelium, which is aseptate and choanocytic. If I talk about choanocytic, we have studied this term earlier as well. So this is for those hyphae. Hyphae are basically just long thread-like structures, and these long thread-like structure which are continuous tube filled with multinucleated condition, multinucleated cytoplasm. So these are continuous tube filled with multinucleated cytoplasm. So the cytoplasm is multinucleated and they are majorly aseptate. Okay, they are not branched. And further on we talk about spores, and spores are produced endogenously. Okay, the asexual spores are produced endogenously here. Are produced endogenously. Endogenously, which means inside the asexual spores. Further on, we are going to study about the examples. Examples include Rhizopus, Mucor, and Ascomycetes, the commonly known sac fungi. Ascomycetes are the commonly known sac fungi, and they are majorly multicellular. All the fungi are majorly multicellular. There is an exception also, which is our yeast or Saccharomyces cerevisiae, and they are saprophytic, decomposers, parasitic, or corpophilous. As we talked about earlier, habitat, like in Phycomycetes, they are obligate parasites. They can be saprophytic, they can be found in that decaying in most soil and damp places, majorly aquatic. The Ascomycete fungi are saprophytic. Saprophytic means they are decomposers as well. They are corpophilous as well. Corpophilous means growing on dung. Growing on dung. If I talk about mycelium, mycelium is septate and branched. Mycelium is septate and branched. If I talk about mode of reproduction, there are two modes of reproduction, asexual and sexual. Sexual and sexual. If I talk about asexual mode of reproduction, the spores are produced endogenously. And sexual spores are produced exogenously. However, in the case of Phycomycetes, there is no mention about the asexual spores, but sexual spores are produced endogenously. And here the thing reverses and the sexual spores are now produced exogenously. Now I'm going to talk about asexual spores. These are conidiophores, and sexual spores are ascospores. Ascospores. Fruiting bodies are ascocarps which produce ascospores. Now we are going to study about the examples. Like founder of haploid genetics. Haploid genetics. If I talk about aspergillus, it is nothing but aspergillus used for the production of citric acid. Aspergillus niger. Further up we have claviceps. Claviceps. Further on, we study about some other examples, which are famous examples like penicillin, the first antibiotic, whose full potentials were studied by Ernst Chain and Harvard Florey in 1945. They were awarded with the Nobel Prize. However, it was discovered by Alexander Fleming. Alexander Fleming. At last, we have yeast used in the production of ethanol, cakes, pastries, bread. Yeah. So that's it about ascomycetes. Now we are going to study about basidiomycetes. Basidiomycetes are known as the club fungi. The club fungi. If I talk about basidiomycetes are nothing but they are the one where sex organs are absent but still sexual reproduction is present, right? And they have a sexual reproduction where asexual spores are not found, but vegetative reproduction is there. And let's begin. If we talk about their habitat, so their habitat is like they grow on soil, logs, tree stumps, are parasites. Soil, logs, stumps, they are parasites as well. So if I talk about the number one, that was our phycomycetes. They are majorly aquatic. They grow on dampened. Yeah, they are saprophytic. Uh, they are also obligate parasites. Then we have had ascomycetes, which are parasites as well, and which are saprophytic and copophilous, that is, they grow on dung. Then we have again saprophytic and parasites. So majorly they are saprophytic and parasites. But if I talk about obligate parasites, they are majorly phycomycetes, as it has been mentioned in the book. And further on, we move ahead with the asexual spores absent. Spores absent. Vegetative reproduction through plasmogamy. Through plasmogamy. If I talk about uh, sexual spores, sex organs are absent here. Sex organs are absent here. And if I talk about sexual spores, sexual spores are known as basidiospores, which grow on basidiocarp, and they ultimately give rise to basidium. And that's it. We have some examples, including agaricus, the mushroom. We have puffballs. We have bracket fungi. We have agaricus. We have ustilago. And we have Picinia. We trust, and ustilago is mud. And agaricus is mushroom. Vegetative reproduction is through fragmentation. Next one, we study about deuteromycetes, where sexual reproduction is not found. It mimics sexual reproduction. That's why it is known as the fungi imperfecti. And when the sexual reproduction of these fungi are found, they are further put into different things, and at last in the um, acidiomycetes or basidiomycetes, and they reproduce only by asexual spores known as conidia. They are known for the mineral recycling and decomposition of litter. So that's it. And they have examples: Alternaria, Quello, Trichum, and Trichoderma. Trichoderma is known for producing cyclosporine, which is immunosuppressive, immunosuppressing in nature. That's it about kingdom fungi, and in the next session we'll study about kingdom Plantae, kingdom Animalia, viruses and viroids, and then our chapter will be finished. So, what we studied today was kingdom fungi. Kingdom fungi, we studied about hyphae. We studied about mycelium. Hyphae is long thread-like structure. Mycelium is nothing but the network of hyphae. Okay? Hyphae we studied about quenocytic. And other one was septate and branched. Quenocytic is multinucleated, long continuous tube with filled with multinucleated cytoplasm. Septate and branched. Phycomycetes have quenocytic, others are septate and branched. If I talk about other thing that phycomycetes have is the production, the spore production. So endogenous sexual reproduction, and if I talk about others, they have exogenous sexual rep and endogenous asexual reproduction. Next we can talk about morels and truffles. Morels and truffles, which are in ascomycetes. If I talk about parasites, phycomycetes are obligate parasites, and others are parasites. Others have parasitism as well, and all are saprophytes. Mycelium expands centrifugally, and that's it for today. Thank you. Have a great day.